All right, it is such a pleasure to be here with you today. My name is Tom Baer. I'm a medical oncologist and I serve as Chief Medical Officer for Multi-Cancer Early Detection at Exact Sciences, and I'm pleased to give you an update on our program in the development of blood-based multi-cancer early detection tests. First, my disclosures. In addition to being employed by Exact Sciences, I'd like to discuss prior research funding when I was a faculty member at OHSU and worked with a number of companies involved in the cancer early detection area, and those are shown here. So why are we all here? Well, I think the fact that cancer is a second leading cause of death in the United States, anticipated to become the top cause of death by 2030, claims the lives of 600,000 Americans and 10 million people worldwide every year, is really why we're so focused on tackling the cancer program. And we believe that early detection of cancer with effective early treatment is a promising way to significantly impact on the burden of cancer. But the state of the field today leaves us with only 4 cancers for which consensus recommended screening tests are available. Those are shown here. And for lung cancer, it's for a subset of individuals at higher risk as a result of smoking exposures. And so that leaves us with 2/3 of all incident cancers and 2/3 of cancer deaths attributed to all the other cancers for which we do not currently have screening tests available. We believe that developing multi-cancer early detection tests is a promising way to address this gap, particularly because developing additional screening tests, one cancer at a time, would be quite challenging with so many cancers contributing to that remaining 2/3. Some of the features of multi-cancer early detection tests that are certainly defining for our test in development, but really inform the efforts of the entire field are shown here. So first of all, we and others are developing blood-based early detection tests that seek to detect biomarkers released from cancer into the bloodstream. The idea here is that these tests, if successful, will markedly expand the range of screening to include many cancers for which no screening is available. And it's critically important that they do so with adequate sensitivity for early stage cancers where interventions can make a difference. So it'll be important that they detect multiple different cancers and detect them at earlier stages. We also hope that developing a blood-based multi-cancer early detection test will enable us to bring screening to everyone who needs it across all populations. And one other design feature that is common and worth mentioning is that these tests are being designed with a priority on high specificity, meaning we are seeking to avoid as much as possible false positives, and in the trade-off between sensitivity and specificity, we're obviously looking at both, but putting a priority on having a high specificity test. These tests are being made possible by advances in the understanding of cancer biology, which has really increasingly described the hallmarks or biologic drivers of cancer, and advances in diagnostic technology that enables us to detect these hallmarks of cancer, these low concentration biomarkers in the blood in a very sensitive manner. And there's a variety of biomarkers that one could examine. Our current effort at Exact Sciences has focused on 3 DNA-related markers: mutations, aneuploidy, and methylation, and also cancer-specific protein biomarkers. Our efforts in this area have been long-standing, have begun a number of years ago now, primarily in the laboratories at Johns Hopkins University, where Bert Vogelstein and that group developed a concept of a multi-biomarker class blood-based cancer early detection test focusing on mutations and proteins. The initial publications from that effort are now more than 5 years ago. Not long after the initial concept, the group carried out and reported the first prospective interventional clinical trial of a multi-cancer early detection test in the United States. And we'll talk more about the results from that trial called DETECT-A in the coming slides. Since then, a lot has happened. The technology from Johns Hopkins became a part of Thrive. Thrive became a part of Exact. And at Exact, the scientists have continued to work to add technologies developed at the Mayo Clinic and inside our own laboratories at Exact Sciences to expand the test from 2 biomarker classes to 4 biomarker classes. And I'll be updating you on the latest developments in that regard. But first, DETECT-A. DETECT-A was the first prospective interventional study. It was really a feasibility trial, and it enrolled 10,000 women, which gives you a sense for the scale required even for feasibility or pilot studies in cancer early detection. Some of the things that were done in DETECT-A continue to inform our approach today. So, for example, an IV contrast CT and PET-CT approach to diagnostic resolution of a positive signal from an MCED test remains the centerpiece of our approach to resolving MCED tests. Some things have evolved. So in the DETECT-A study, the blood test required a second blood draw for a confirmation of a positive signal. That is not part of our current test, nor do we anticipate doing that in the future. We've been able to combine all the analytics, including confirmatory analyses, into a single blood draw. DETECT-A, as I mentioned, enrolled 10,000 women. They were aged 65 to 75. And the only other eligibility criterion was that participants could not have a prior history of cancer. The core results from that trial are summarized here and have been published in DETECT-A over the first 12 months of observation. 24 cancers were diagnosed as a result of standard of care screening tests. These women continued to get routine standard of care screening tests as part of their care. 26 additional cancers were detected directly as a result of the blood test, and 46 cancers presented clinically. So a single prototype of an MCED detected as many cancers as all of the screening tests available today as standard of care combined in this 10,000 participant study. The distribution of stages of cancers detected on the MCED is shown on the right, and you can see that all 4 stages were represented. Not all cancers were early stage, but the test was capable of detecting a number of early stage cancers, and that's going to be important for outcomes, which we're going to discuss shortly. This slide shows you the complete detail of every cancer diagnosed in this study. And what you can see is first the listing of the cancer organs of origin or types. The first column shows cancers diagnosed as a result of the MCED test. Second column, those as a result of a screening test. And lastly, cancers detected clinically. And I'll make a couple of observations here. First, you can see the stage distribution. You can see that for the blood test, what stands out is the number of ovarian and lung cancers detected. For the standard of care screening, of course, what's illustrated here with the gray boxes is that most of the cancer types that we're seeing are not addressed with standard of care screening, as we know. And on the right-hand side, you can see the clinically detected cancers. The single most common was early stage endometrial cancer, which is not surprising. That presents with vaginal bleeding in postmenopausal women at a stage where one would not necessarily expect it to spill a lot of DNA and protein biomarkers into the blood. I mentioned already that DETECT-A defined our diagnostic resolution strategy for MCED. In DETECT-A, we used an imaging-based approach to search for the cancer. It involved an IV contrast CT and PET. This was all done at the Geisinger Health System, which had the capability of combining an IV contrast CT and a PET scan into a single imaging session. That is not something that is typically done in the United States in routine clinical care, where most PET scanners do not support high-resolution IV contrast CT. So going forward, we've separated those two into separate image acquisition sessions, first an IV contrast CT, then a PET scan if necessary, but the basic approach remains the same. This imaging-based approach correctly localized all 26 cancers that were diagnosed in this study and resolved the false positives effectively, and I'm going to show you some data on that in a moment. So what we've been able to do recently and update the results from this study at the ASCO meeting not long ago was to take a look at the false positive patients and the true positive patients through the lens of long-term follow-up. By virtue of having the first prospective study of an MCED, we now have the longest available follow-up in the field. We've got 4.3 years of median follow-up on these individuals, and so we can look at their cancer outcomes over several years. This is a complete accounting of the 134 patients who had a positive MCED test in the study. You can see that 7 were not recommended for further follow-up. These were by and large individuals with a protein biomarker elevation that was understood and known about prior to study entry and explained by non-cancer causes. 3 individuals had precancerous conditions diagnosed, and that left 26 cancers diagnosed, so-called true positives, and 98 false positives. And those are the 2 groups that we've been able to look at with longer-term follow-up. So let's take a look at those longer-term follow-up data. First, the false positives. There were 98 individuals with a positive MCED test and a negative imaging and clinical evaluation, and no cancer diagnosis during the first 12 months of observation. This is an area that there's a lot of interest in. There are a lot of questions that I encounter and many of us encounter about what is the meaning of a false positive. Does that identify patients who may have cancer that we weren't able to find? Does it identify patients at an elevated risk of cancer that need to be followed closely? Is it going to leave folks worried and anxious about their cancer status? So having long-term follow-up on these patients is really critically important to helping with these questions. What we found in our follow-up examination of their records is that with 4.3 years of follow-up, 96 out of 98 of those patients remained free of cancer. That's 98%, meaning that 2% developed cancer over a 4-year period. That's about a 0.5% per year rate of cancer, which is a little bit below what you'd expect in this age group, a little bit below what we saw in the first year of the study, when it was just under 1%. So what we're able to conclude from these results is that with our test and our diagnostic resolution strategy in this feasibility trial, we're not seeing a residual elevated risk of cancer. We're seeing a risk of cancer that is back to baseline, if not a little bit lower, and is unlikely to require things other than routine clinical care that these folks would receive regardless of an MCED test result. The 2 cancers that were diagnosed are shown here. They were both diagnosed between 2.5 and 3 years following enrollment. One was a very small early stage breast cancer. The other was a stage 3C aggressive ovarian cancer that, despite surgery and chemotherapy, claimed the life of that woman within a year of diagnosis. We can't conclusively determine whether these cancers were missed by the MCED test or they were new cancers that occurred later. But the fact that they were diagnosed between 2.5 and 3 years later, and their clinical characteristics raise a suspicion that they're new interval cancers and were not present at the time of MCED testing. The breast cancer is so small that it seems unlikely that it had been present for many years, and the ovarian cancer was very, very aggressive. And similarly, one might think it was not present 3 years earlier, but we don't know that for sure. regardless, the rate of cancer risk following a false positive is quite low. Now, turning to the true positives, the cancers that were diagnosed. Remember, we do this work in order to diagnose cancer earlier so that interventions can be deployed that are effective and lives can be saved. So what happened with these folks over the longer term? On the left-hand side, you can see a reminder of the stage distribution. There were 9 stage 1 and 2 patients, 8 stage 3 patients, 9 stage 4 patients. Amongst those that are stage 1 and 2, those that were treated, which is 8 out of 9, are all alive and cancer-free as of last follow-up. There's one patient who chose observation is also alive. Those who were diagnosed at stage 3 did not have as favorable outcomes, but still about 2/3 of those individuals are still alive, and a little bit over 1/3 are alive and cancer-free. Qualifying for surgical therapy was an important predictor of favorable outcomes. Of course, this is highly correlated, though not completely overlapping with early stage diagnosis. 14 out of the 26 patients, or 54%, were treated surgically, and 86% of those individuals are alive and cancer-free at last follow-up. Now, I know to this group, lung cancer is of particular interest, and we've not presented these data before, so these are new data being presented today. We looked at the 10 lung cancers diagnosed as a result of the MCED test, and you can see that those that were diagnosed at stage 1 or stage 2 remain alive and cancer-free. Those that were diagnosed at later stages have passed away. So in conclusion, what we're seeing is that our early version of the MCED test more than doubled the number of cancers that are screen detected. It was capable of detecting cancers early enough for effective treatment, and a number of these individuals, including 3 individuals with lung cancer, received surgical treatment or radiation treatment and are alive and cancer-free 4 years and beyond since enrollment in this study. And what we're seeing is that the imaging-based diagnostic strategy that we've deployed and plan to continue with did a good job of identifying the location of the cancers and did a good job of reassuring those individuals who have a false positive result, leaving them with a low remaining risk of cancer over the ensuing 4 years. So now, where are we with the development of our test going forward? As I mentioned earlier, our focus has been on improving test performance with a particular aspiration to improve the performance for early stage cancer detection. That is obviously the goal of cancer screening. I've just shown you data that when cancer can be detected early, outcomes can be really remarkably favorable. So we want to make the test as sensitive as possible for early stage solid tumors. And to accomplish that, the team has added 2 additional biomarker classes, methylation and aneuploidy, to the 2 biomarker classes, proteins and mutations, that were used before and that were responsible for the results that I just shared with you. We reported the case control analysis results for this multi-biomarker class test last fall, looking at 15 different cancer sites using about 4,000 samples that were divided into training and validation set, where the test was really developed, designed, and locked down, and an independent test set where its performance was then evaluated. And so here's what we saw in these data, looking at all of the cancer types that we examined. The test had an overall sensitivity of 55% when we used 3 biomarker classes and 62% when we used all 4 biomarker classes. So about a 7% improvement in sensitivity with the 4th biomarker class. Specificity was 99 and 98% respectively. The test did detect all 15 cancer types that were tested, And I want to make the editorial comment here that the test was not designed for those 15 cancers. This test and many others in the field rely on the detection of shared cancer signals that we would expect to see from a broad range of cancers. We just happen to have tested it against a panel of 15 cancer types, but we do expect the test to be capable of detecting a broad range of cancer types based on the biologic contents of the test. Most importantly, from our perspective, that improvement in sensitivity was concentrated in the early stage cancers. So for stage 1, we were able to achieve a sensitivity above 30%, and for stage 1 and 2 combined, above 40%, for stage 2 alone, just above 50%. These sensitivities, when compared to single cancer screening tests like Cologuard or colonoscopy for colon cancer, mammography, low-dose CT, they're not as high as those dedicated single organ tests that interrogate that organ directly and not through the blood. But for a multi-cancer test that relies on blood markers, these levels of sensitivity for early stage, from our perspective, are really encouraging, give us a lot of confidence going forward. And from a public health standpoint, because these sensitivities apply to a broad spectrum of cancer, they have a substantial opportunity to enable early detection and drive down the mortality and morbidity from cancer in the future. Now, turning to lung cancer, again, we've not disclosed these data before, but especially for this meeting, we dipped down into our case control set and focused on lung cancer so that you could see the results in lung cancer. There were 94 cases of lung cancer in the test set. You can see the distribution by stage: 19 for stage 1, 15 for stage 2, 30 and 30 for stage 3 and 4, respectively. The overall sensitivity for lung cancer was 62%, or nearly 63%. And you can see the sensitivity by stage. For stage 1, it was 10%. For stage 2, it was 66%, and then it was higher than that, as you'd expect, for stage 3 and 4 cancer. So these are early days, not a huge number of cases, a relatively encouraging sensitivity for stage 2 lung cancer. We'll see about stage 1 as we continue to work on it with the next case control study. But obviously, that's not going to be at the level that you'd expect from a dedicated imaging-based screening test that looks at the lung directly. So what we took away from this developmental program is that a multi-biomarker approach, including 3 and 4 biomarker classes, really yields encouraging results with regard to moving forward with an MCED program and aiming to really make a dent in early detection and cancer burden in the long term. We evaluated cancer types that represent about 75% of all cancer deaths, and the test detects all of those. We do expect it to detect others as well. And in the next case control study, we'll be looking at an even broader panel of cancers. Overall sensitivity at 62%, along with notable improvements in sensitivity for stage 1 and 2 cancers, were encouraging to us. And stage 2 lung cancer sensitivity also looks quite encouraging, not as a replacement, of course, for dedicated screening, but as a complement. So we envision the future of screening as a world where multi-cancer early detection alongside proven dedicated single cancer screening tests that exist today markedly broaden the reach of screening and enable the detection of a broad range of cancers, creating the opportunity for early intervention and hopefully resulting in a significant reduction in advanced cancer and in cancer morbidity and mortality. In order to get us there at Exact Sciences, what we need to do is to finalize our assay, and we're doing that in the next generation case control study called ASCEND-2. This is sort of the the penultimate case control study for us. It is contemporarily collected samples using LB-GARD tubes, which are the optimal tubes for our biomarkers that we intend to use in the assay going forward. Samples are collected immediately after diagnosis, prior to initiation of any treatment. Broader range of cancer types, larger number of sample types. So really a very robust case control study that we are working on right now and anticipate reporting the results from in the fall of 2023. After that, we intend to initiate a pivotal clinical trial. This will be a very large prospective interventional randomized clinical trial. We intend to conduct the largest randomized study of an MCED test in the United States. It will have a control group. It will have long-term follow-up and a broad range of endpoints. It is being designed with the advice of the FDA and the goal of getting a test through FDA approval. We believe that in the United States, an FDA-approved test is essential for broad availability, for equitable availability, and is really a foundational goal for broad acceptance by payers, and therefore a broad opportunity to impact on the cancer problem. In order to accomplish this program, we're not working in isolation. We're working with a lot of folks in different areas that are relevant to the MCED program. We're naturally working with a great number of investigators as we get ready to launch our pivotal trial, a number of hospitals and health systems and institutions and research institutions, also the National Cancer Institute, the FDA. We're interacting with a lot of advocacy groups that are working hard to educate patients, the community, physicians about the opportunity of MCED, influence relevant policy. We're also talking with policymakers where we need some legislative adjustments to policy in the United States to enable access to this new category of screening tests. So a lot of folks are involved here in advancing the goals of MCED development. And so in conclusion, we believe that MCED testing has real potential to be a paradigm shift in cancer screening, primarily through expanding the reach of screening to the many cancers that are currently unaddressed by screening. That includes, as you know, some lung cancers, for example, lung cancers in non-smokers or light smokers who may not qualify for CT-based screening today. We've shown in DETECT-A that MCED testing can double the number of screen-detected cancers, including those with no recommended screening, including lung cancer, complementary to available screening. The majority of the cancers detected were either localized or regional. And for those that were localized or those who qualified for surgical therapy, we now have 4-year outcomes that show a real impact of early intervention, with half of all the patients who had a cancer diagnosed still alive and cancer-free beyond 4 years. We've shown that false positives, when resolved with our approach, have a low residual risk of cancer. And if confirmed, these data would provide evidence that they do not need to be concerned about residual cancer risk beyond what any average risk individual would think and be concerned about. They would not appear to require any special follow-up beyond continuing routine healthcare, including standard of care screening. And then, in our developmental program, we've shown the promise of moving beyond the 2 biomarker classes that were tested in DETECT-A to a 3 and 4 biomarker class design. That's resulted in improvements in sensitivity for early stage cancer and will serve as the basis for our further development and a large pivotal randomized trial. And with that, I thank you very much for your attention. Really appreciate the chance to present to you today, and wish you a wonderful conference. Thank you very much.